Good afternoon. Thank you very much. Good afternoon, esteemed guests, colleagues, and students. Welcome to the 2024 Future Leaders Symposium. My name is Soy, and I'm a research officer in the Blood Cancer Research Group at Mater Research. The Future Leaders Symposium is a celebration of the great achievements of our higher degree research students and early career researchers at Mater Research. Today, we have over 160 registrations and guests, including Mater Foundation supporters and Ohio Schools students and staff. Each year, we come together to celebrate the outstanding contributions of our future leaders in biomedical and clinical research towards better health outcomes for the community. The Future Leaders Symposium provides a forum to learn about research with impact at Mater and within the community at large, and also to discover how our future leaders are on their journey to address some of the world's biggest health challenges and unmet needs. Some housekeeping. In case of fire, the public address system announcement will be made asking you to exit this particular auditorium. The main fire exits at TRI are also the entrances that you came in just now. So restrooms are located on the ground level outside the auditorium, left to the TRI main entry. Smoking is not permitted within 5 meters outside the fence line of the TRI building. And all presentations will be recorded today. By entering this event, you agree to being filmed or photographed, which may be used for marketing or promotional purposes. Q&A sessions will be limited to 2 minutes only. Presenters are requested to discuss their research and respond to questions during afternoon tea and the networking session. Now I would like to invite Davina Smith from the Indigenous Health Research Group for Acknowledgement of Country. Davina Smith is a Gurang South Sea Islander woman who recently joined the Indigenous Health Research Group at Mater Research. She brings leadership and expertise in working collaboratively with a focus on Indigenous communities' strength and resilience and the ability to provide cultural mentoring and support. Davina led a team implementing strength-based birthing and health services designed to support the health and well-being of families in Southeast Queensland during the perinatal, birthing, and early childhood stages. She has a Bachelor of Psychology in Behavioural Studies. Davina?
Good afternoon, everyone. Just like to say it's so good to see you here this afternoon at the Future Leaders Symposium. And it's my privilege and my pleasure to acknowledge the traditional custodians of the Yagara and Turrbal peoples and acknowledge their contributions, to not only culture, but still land and seas, and just pay my respects to the elders past, present, and the Indigenous representatives who are also in the room. And just like to also say congratulations to the researchers who are having their work acknowledged today. Thank you.
I would like to open the symposium now with the John Prins Oration. Professor John Prins was the CEO of Mater Medical Research Institute in 2009. He became the director of MATA Research Institute, UQ, in 2013. In 2018, he was the acting group CEO at MATA, and consequently, he's the executive director of Health Translation Queensland in 2023. He is a clinician scientist, clinical training in endocrinology, based in Brisbane, and then did his PhD at UQ. He has fostered research excellence in the MATA research, ECR, and PhD cohort, and it had major contribution to the recruitment of researchers and integration of research and clinical activities. I would like to welcome Associate Professor Jacob Begun and Ms. Raya Brunello as the plenary speakers for the John Prins Oration. Associate Professor Jake Begun leads the IBD group at MATA Research. He's the Director of Gastroenterology at MATA Hospital Brisbane and international expert in the IBD field. Associate Professor Jake Begun's research exemplifies MATA values with his collaborative approach, spanning across patient care, clinical trials, clinical research, and translational research, focusing on the unmet health needs in the IBD patient population, delivering benefits to the MATA community as well as beyond. He has established one of the biggest IBD biobanks in Australia with over 500 tissues, specimens from a very well-characterized cohort of IBD patients at the MATA Hospital. The MATA IBD Clinical Trials Unit is the most prolific IBD-focused clinical trials unit in the country with extensive experience in running industry or investigator-initiated clinical trials. Ms. Raya Brunello has been Associate Professor Jacob Begun's patient for many years now and will be speaking today with Professor Jake Begun. She will provide a perspective on living with IBD and participating in a clinical trial that Associate Professor Begun's team are running at the MATA Hospital called Stop It, a trial examining the efficacy of a drug called tofacitinib in chronic pouchitis. Thank you, Jake.
I think I'll start off with the podium mic and then we'll put them up there. So thank you very much for that kind introduction. It's a pleasure to be here. And I guess I'm one of the people that John actually recruited here. I came in 2013 to the MATA Research as one of these clinician scientist type people. And today I'll briefly introduce IBD and the trial, and then I'll ask Raya to come up and share her perspective with us, and then maybe finish off with some of the preliminary data that we have from the trial so far, because it's very exciting, I think. Okay, perfect. All right, so realizing that not everyone here is an expert on inflammatory bowel disease, I'll just briefly introduce ulcerative colitis, which is an inflammatory condition of the colon. I'm a gastroenterologist and spend half my time, a little bit more, in the clinic taking care of patients with this condition and Crohn's disease, the other half in the lab. And ulcerative colitis and inflammatory bowel disease on the whole is a disease of unequal distribution around the world. This is a heat map of the world, showing where the hot spots of inflammatory bowel disease are. And you can see Australia down there at the bottom right is one of the highest incidences of inflammatory bowel disease in the world. And what we've noticed around the world in epidemiological studies conducted here and elsewhere is that the incidence of inflammatory bowel disease is rising. The first case of ulcerative colitis was described by Sir Walter Banks in 1859 in the UK. And since then, we've seen the incidence increase in the Western world. And it was thought to be just a disease of the developed world. But now we're seeing another epidemic taking off in the newly industrialized world, Southeast Asia, South America, Africa. And on the left-hand side, you can see the incidence of Crohn's disease and ulcerative colitis going up and up and up. Good time to be in the business of IBD, I guess. So why is this increasing incidence happen? Well, a lot of effort has been put onto genetics of IBD. And in fact, IBD is one of the poster children of GWAS early on. We've discovered now over 500 genes in this complex polygenic architecture of IBD that contribute to increased risk of inflammatory bowel disease. But genetics can't explain this increasing epidemiology. And so we know there are other factors, and mostly we look at the environment. And one thing that comes out quite strongly from the epidemiological studies is that what we eat is affecting our guts in not so great ways. So ultra-processed foods, high refined sugars, and additives that happen in our food supply, as well as vaccination, the hygiene hypothesis, et cetera, lead to a condition where our microbiome, those bacteria that inhabit our gut, are dysbiotic. They're not quite right, and they're triggering this immune dysregulation, which then causes sort of an auto-inflammatory state, and that is IBD. And so this is our current hypothesis of what's going on. So with ulcerative colitis, which is this chronic inflammation of the colon, first of all, a note about incidence. We think that there's about 100,000 people in Australia with IBD, and just under half have ulcerative colitis, so more than 40,000 people with ulcerative colitis. And over the course of a patient's lifetime, up to 20% in some studies will eventually have some complication of their disease, either uncontrolled inflammation that we can't control with our current medications, or they can develop cancer, or other complications that require colectomy. Colectomy means we do a surgery that removes your entire colon because we can't treat it with medicines anymore. And in that setting, we'll often do a restorative procedure called a pouch. And that's what this talk and this study that I'm going to describe is about. So I was told not to use real surgery photos because they might be disturbing. So we have cartoons instead. On the left-hand side, we can see the natural anatomy with the stomach, the small intestine, and the colon. I'm just going to pop on a laser pointer here. This is your colon running out here. It's about an 80-centimeter long organ. That's what we look at when we do colonoscopies. And in ulcerative colitis, this is the organ that gets inflamed. And so we can remove the colon in a procedure called a proctocolectomy. And then we construct a neo-rectum out of a pouch made from small bowel. So the ileum is constructed into this little pouch. In the first stage of the procedure, we have a stoma. So all of this contents go out through the stoma so that this pouch can heal after you form it. And then in the second stage, we close that off, and then we're left with this pouch, which is great. This restores continuity. You don't need to wear a stoma bag anymore. And from the outside, at least, everything looks normal. And it's great when pouches work well and things are nice and comfortable, but sometimes it doesn't go so well, and things can also happen with these pouches. And the most common complication of pouches is a condition called pouchitis. We're gastroenterologists and not very sophisticated, so we just add itis on the end of anything. So colitis, pouchitis. So that's when you have inflammation in the pouch. And so here we see what looks like a J pouch. Again, not creative. It looks like the letter J, so we call it a J pouch. And you can see that there are these little ulcerations that can happen within the pouch, and that's de novo inflammation that happens within the pouch. And when that happens, people have a variety of symptoms, urgency, bleeding, pain, lots of things that can be a problem. Interestingly, there seems to be a bacterial etiology to this because it responds, initially at least, to antibiotics. But then sometimes people become antibiotic dependent or even antibiotic refractory, where the antibiotics don't work anymore. And up to 20% of people get chronic pouchitis. And there's only one approved therapy, and it actually was just approved last year, and that's vedolizumab. And it's about 60% effective, which in IBD world is not so bad, actually. But that leaves a major unmet need. And that's where this trial comes in. And this trial was of a new molecule called tofacitinib. Tofacitinib is a JAK inhibitor. JAK inhibitors are all the rage now in immune diseases, immune-mediated inflammatory diseases. They inhibit JAK kinases. Janus kinases, or just another kinase sometimes referred as. And inhibiting these kinases can inhibit STAT phosphorylation and activation, which then inhibits downstream transcription of pro-inflammatory cytokines. They're very nice. They're small molecules. They're taken orally compared to vedolizumab, which is given as an intravenous infusion, and they work in ulcerative colitis. And so we thought, well, maybe we can look at tofacitinib in the setting of chronic pouchitis, because that was an unmet need. And we started a trial. Of course, when you do a new trial with a drug, you always have to come up with a clever name, and it usually involves various letters in the word of your title. And so we managed to call it Stop It, and that's our little logo over there. And this is a national investigator-led, moderator-led, multi-center open-label induction and randomized double-blind placebo-controlled withdrawal trial. That's a mouthful. What it looks like is that people come in, and we thought it would be unethical to randomize people to placebo coming into a trial. So everyone in this trial gets drug up front, and we can see if they respond or not. Responders are then allocated in a randomized double-blind fashion to either maintenance with the drug or placebo, and then our readout is at 24 weeks, thinking that if you have initial response to drug, randomized to placebo, lose response, that'll give us the power we need to show it's effective. Some people take a little bit longer, and so if they don't have a response at the end of 8 weeks, they get another 8 weeks of treatment, and then they can go into randomization. And then afterwards, there's an open label extension for people who are doing well. So I just wanted to introduce the the topic and the background, talk a little bit about the trial, and then I think now I'd like to ask Rhea to come up to the front here so that we can just have a little chat about what life is like from a patient perspective, and then I'll come back and talk about some of the results from the trial. So Rhea, if you want to join me up front. So, testing 1, 2, 3. Yes? Okay, good, perfect. Have a seat. So Rhea's been a patient of mine for a little while now. Actually, she previously saw Professor Florin, who was also a member of Modern Research before I arrived. And she has had a diagnosis of ulcerative colitis for a long time. So I thought that she could share with us just a little bit about when you were first diagnosed, which was a few years ago now.
Yes, that was when I was 20. Well, I just turned 21 in 2001. So I was in my last semester of uni and about to go on a bit of a gap year, and became quite unwell with some symptoms, which was then exacerbated by the fact that my brother had eaten a bad Chico roll and got Campylobacter poisoning. So he also had sort of similar symptoms to basically excruciating bowel pain and PR bleeding and weight loss at the same time that I did, but turns out they weren't related. So by the time I got to hospital, and also I'm from a medical family, so you have to be dying before anyone pays any attention to you. So I was basically, by the time I ended up in a hospital where somebody went, oh, there might be something else going on here, I was quite toxic, and very quickly basically escalated to have my bowel removed. That didn't happen. We sort of did some treatment and things improved a little bit, but yeah, after a little while of being very, very unwell, I then was diagnosed with having chronic ulcerative colitis, although the question mark, I think it's probably not ulcerative colitis, something in between. It's never ever actually been exactly defined what it is, because it's a bit different to usually what defines UC. But yes, so that then began a period of essentially a disease that controlled my life, really. So I didn't end up going overseas. I did end up finishing my degree for a while, and spent a lot of time in and out of hospital, lots of different doctors. But essentially, you know, you go through periods of being well-ish versus periods of being particularly unwell, which, you know, the question that defines that is how many times a day you're going to the bathroom. So when you're really unwell, you might be sitting on the toilet all day because the pain's that bad, and you're, yeah, just not much you can do to control it. So I definitely didn't really respond to much. I've been on every drug that there's possibly that you might want to take for ulcerative colitis.
Absolutely. And
so
that was about, I think, 7 years or so between diagnosis and then
yes,
ultimately
so eventually
surgery.
after exhausting every possibility, I decided to proceed with having surgery. In the lead up to that, I had got married and I had 2 children. And interestingly, each time I was pregnant, my disease seemed to disappear a little bit. But as soon as after I'd had the baby, and a few months after having a baby, I would be back to being quite unwell and in hospital. Yeah, after my second child, I decided that that was that, and proceeded with having.
And how was your quality of life like after surgery compared to before surgery, would you say?
After surgery, it was, well, after my, so I had a total colectomy and a temporary stoma. So, and yes, it was actually better having a stoma than having ulcerative colitis. So immediately I felt better because systemically my bowel was removed. And so, yeah, and then once I had the procedure to join everything back up again with the J pouch, again, it was better because I didn't have that horrible organ still inside me. But I, within a few months, obviously then developed pouchitis, which did respond to things possibly a little bit, but not so much.
And in fact, Ray actually was treated with vedolizumab when I was taking care of her, which we initially had a response, but then lost response, which unfortunately happens a lot with IBD therapies. And then I said, oh, Ray, I have this great trial for you that one of my PhD students is doing. Would you consider participating? And fortunately, Tim had already primed you for participating in trials, I think, so you were kind of open to the idea.
I had done a few trials.
What was it like as a patient participating in a clinical trial?
Well, I think when you've got a disease that there is no cure for, you essentially, you know, my first question to the doctor would always be, are there any new trials? what's happening, you know, what can I go on? So I was always very enthusiastic about participating in trials because I mean, whether it was going to be a cure, but possibly something that would improve my day-to-day functioning. Yeah, and I think now that I'm a bit older, when I hear of anyone who's younger or my age that has this disease, I immediately feel for them because it does ruin your life in a way. So it's very antisocial disease, and so a trial was, yeah, I always jump at doing something, especially for pouchitis, because I think ultimately if you can control that, it's a pretty good solution to the current problem.
And Ray was fortunate she responded quite well in the clinical trial and made it all the way through maintenance. And in fact, not on any medication right now and doing well. We might have reset things for a little while, which would be good. And here we have a captive audience, Ray, and just in the last minute or so, is there anything that you would like to tell all these budding researchers for what they should be looking at or trying to do as they go forward on their careers? Words of wisdom.
Definitely gastroenterology.
There you go.
Specifically pouchitis. But I mean, anything in that area, and always think that, I mean, that's ultimately it. Whenever I hear about anyone doing new research, I just feel positive for anyone that's got that disease, and the idea that there might be, obviously, if you're not preventing it, how we're treating it. And it's very hard hearing that what you've got there's no cure for what you've got. So once that sort of sets in as a real reality, your next thing is, well, how am I going to treat it? And yeah, so yeah, please do your research.
Oh, super. Well, thank you very much, Ray. I'm going to ask Ray to sit down now, but then I'll ask her at the end to come back up again for questions so we can ask all questions around them. Thanks so much. All right, that was super helpful to get a perspective about what it's like coming into trials. I just want to talk a little bit about the preliminary data that's come out of the trials, and then we can take some questions at the end. So we're about halfway through recruitment for the trial, but we have the induction data, which is unblinded, so we know that patients are on drug, and we can see how they're doing. So of the 42 patients that we have in the trial so far, like I said, about halfway through recruitment, the majority were naive to vedolizumab, and it's important to know if they've responded to vedolizumab in the past because we want to have something that we can use after Vita, which, as I said, is an approved therapy. The median age of patients in our trial was 37. The slight majority actually are female, although it doesn't necessarily reflect the disease, which is pretty much 50-50. When you go down to the bottom there in terms of indication for surgery, about half of patients had their original pouch surgery because of medically refractory disease. About 17% had it for dysplasia or cancer, and about a third of patients had it for acute severe colitis. That's usually when people come into the hospital very, very sick, and during that hospitalization, they're so sick that we have to take their colon out emergently. This is the primary efficacy data. So we see that at week 8, we have about 50% efficacy of tofacitinib in patients who have chronic pouchitis, and that if we do that extended induction out to week 16, we see that the overall response and remission rates raised to over 60%, which is great. Vedolizumab had slightly lower rates of response compared to vedolizumab naive patients at the 8-week mark. Actually, they caught up at the 16-week mark. On the right-hand side is the quality of life. We think quality of life is very important to our patients, as well as objective markers of disease. And we can see that everyone's quality of life improved after tofacitinib if we look at these right 2 columns, but patients who were responded had a bigger response in terms of quality of life measures, in terms of bowel symptoms, systemic symptoms, as Ray was explaining, emotional symptoms, which can go along, as you can imagine, when you're sick, and as well as social functioning domains. So it appears to be effective at inducing remission in chronic pouchitis. We see the remission rates that I mentioned. It's similar to patients who have previously been exposed to vedolizumab, and I didn't show safety data, but it actually looks very similar to the RCTs that have previously been done. But this is also a group of people who are interested in translational science, so I want to mention that as well. So shown here is, state-of-the-art Mater labs. These are actually Sister Mary, Conrad, and Agnes around 1960 in Mater pathology labs. But of course, today's labs are a little bit more futuristic, and we can take on a little bit more. So we actually do have a lot of biospecimens that were collected as part of the Stop It trial. So patients who were participating at the Mater, which is about half the patients, we managed to get biopsies from before they start therapy and after 8 weeks of therapy, and we get a variety of biopsies and material. One of the materials we get is blood, and in the lab, we're able to take that blood, separate it out, grab the white blood cells here, PBMCs, look at T cell subset numbers, isolate out memory T cells, which are supposed to be important to the function and mechanism of tofacitinib, and also look at total PBMCs and stimulation and looking at cytokines. And our question was, can we predict people who would respond based on biomarkers that we can find in the blood? And shown at the top left here, so we do lots of different assays. We look at lots of different cytokines. And shown at the top left here is a unsupervised analysis, or sorry, supervised analysis, looking to see if we can tell the difference between responders and non-responders. In blue are the responders, in gray are the non-responders. And you can see that we can tell the difference pretty well using a variety of different things, including the CD4 count, TNF alpha levels, and the response to tofacitinib when you add them to the PBMCs, IL-17, IL-10, and IL-22. And we think that we're coming up with a signature that can tell us who might respond to this therapy, which would be very important if we go ahead and use it in clinical practice. We also, though, in addition to blood, have tissue samples, which is a really unique resource. And we're looking at lots of different things with tissue, including growing up bacterial cultures from the tissue, looking at RNA expression in the tissue, but also looking at bacterial DNA. And I'll just present the bacterial side of things to start with. And what we noticed as a first pass is that patients who have higher microbial diversity, either by the Shannon index or the species richness, so that's the evenness and the richness of the species, people who have a bit more or a bit less dysbiosis, a bit more microbial diversity, do better and are better response to tofacitinib. But interestingly, we don't see much of a significant change yet with treatment, unfortunately, but we do see this characteristic in the beginning. And also when you look at individual patients and their 16S, this is very messy graph, only to show that there are differences that we're starting to see, as you can look at the p-values in the ileum. So that's actually upstream from the pouch in patients who are responders versus non-responders. So there might be a microbial signal of response and non-response in patients as well. So I don't have a lot of time, but I'll just summarize that functional assays of immune cells reveal differences in cytokine profile, responders versus non-responders at baseline and at V2, which could be useful predicting response. And also about the microbial diversity that seems to be associated with responses to tofacitinib. And hopefully one day we can put this together as a biomarker to predict who might respond better to tofa, who might respond better to veto, and have a rational basis for treating patients, which would be fantastic, because right now we just do trial and error. So if I was just to then quickly summarize the whole talk, performing investigator-initiated clinical trials can be incredibly rewarding, but they can also be very challenging. Recruitment for trials is challenging. Managing things during COVID is challenging, for example, and then trying to coordinate everything and fund everything, it's hard. But coupling clinical trials with translational research, which is one of the things that we can do at this center and at Modern Research, provides a wealth of rich data to answer important biological questions, and that we're really ideally positioned. And I guess for people in the audience, I would just encourage you to remember that this is a long haul, and sometimes you can be at the beginning of that path and it can look like a very long way at the horizon in the distance, but it's really worth it to take that trip. And I would also say to enjoy the journey along the way. So finally, I'd just like to acknowledge the people who did a lot of this work, including Dr. Emmy Koo, who's this PhD. She's a gastroenterologist at the Mater Hospital and is doing her PhD on this trial. Dr. Julie Davies, who is an ECR in my lab, who did a lot of the translational work. This was actually the meeting where we started sketching out what the translational elements of the trial would be. This is the entire IBD translational research and clinical trials team. We were funded, fortunately, by Pfizer, provided the drug they make, tofacitinib, Celltrion Fellowship for Emmy, and the Mater Foundation as well. It's part of the Australia-New Zealand IBD Consortium, of which Mater is the lead organization. And of course, I'd like to thank all of the patients who participate in this trial and all the trials that we run at Mater, and thank you for your attention. I think I'll turn this back on and see if there are any questions. Are you happy if I lead questions? Is that good? Any questions from anyone before there's one right here from Lisa.
Lisa.
We're in good shoes to run? Yeah. Do you want to come up too, Ryan?
Thank you. Really, that was very interesting. And this could be a question for either of you. But firstly, congratulations, Jake, for what you're doing, and thank you for measuring psychosocial aspects. The question is around the extra-intestinal symptoms and signs that a lot of people with inflammatory bowel disease experience. What is happening with those? Are you measuring the outcomes for those in this study?
Super. I'm going to answer a little bit, and then I'm going to let Ray answer as well. So for those of you who don't know, Lisa's conducting a trial right now looking at fatigue and inflammatory bowel disease, because this is one of the most debilitating symptoms. The classic extraintestinal manifestations of IBD include inflammation of joints, of eyes, of skin manifestations. These are the other organs that get associated, but also impacts on quality of life, which include big impact on mental health, and also fatigue comes out in all the IBD trials as one of the most difficult symptoms to treat associated with IBD. And I thought that maybe, Ray, you could comment on, apart from gut symptoms, what are the other symptoms that you think are really important to look at in IBD?
Well, physically, yeah, definitely it's a systemic disease, so everything leads to something else. I mean, I've always had low iron. When I've been really unwell, everything else is affected. And obviously, if you're on a drug, you've got those side effects. But then also, you know, I mean, eating is difficult. Food, what you're doing, at times you basically avoid eating because it's too painful. But, you know, the way you're trying to treat your gut in terms of microflora and all of that stuff, yeah, that's that side of it. You know, there's lots of research into that area. I've tried every possible combination of diets and not diets, and what you're eating and what you're not eating and all of that stuff. But the associated things, it's particularly antisocial. I would say that I was clinically depressed for a long period when I was younger, just managing the disease. You know, I've also had, side effects of, in terms of pain-related side effects, if you're on painkillers, the management of that. Yeah, I think, yeah, it changes your whole life. So it's definitely, you know, the gut, brain, all of that mental health wise, definitely. There's seems to be a lot of talk about that now, and I absolutely agree that there's that issue there. So, but I mean, with any chronic disease, it is mentally very challenging. And, you know, I spent a lot of time, I think, in denial. I just tried to ignore the fact that I had a chronic illness to manage it. But yeah, that wasn't possibly the best way to go about it.
And to answer your question, we are measuring a lot of, we have a lot of standardized survey instruments. Thank you for filling out all the surveys to look at these aspects. So I think they're really important. Even when I can get people's gut healed, sometimes they still have debilitating fatigue. Brain fog is a common symptom that I hear about, and then chronic pain. I think these are some of the really challenging aspects of chronic diseases to deal with, even after you get the gut inflammation under control. I think Ali has a question too.
Yeah, Ray, thank you so much for sharing your journey with us. Jake mentioned that it's really tough to recruit into the clinical trials. And so, but you had mentioned that it's something that you were really keen to find to try and help. So what was your experience of part of the clinical trials that you would think would be a barrier that might prevent other people from wanting to engage?
I have done a lot of trials. I think possibly being associated or at a hospital where there's things happening in the specific disease that you've got is helpful. So I ended up where I ended up for that reason. But I think I don't live in Brisbane, but so I travel. I mean, I'm prepared to travel, and it's time and effort and having procedures where you might have a day out of your life to come up here and have a scope, and also access to drugs, you know, getting all of that medication and things like that, the way that's happening. So, I mean, it can be, you know, often those costs for the participant aren't covered. So, yeah, I think access, the way that you can actually access it is, can be quite difficult. So, you know, I would say a lot of the time I might have spoken to people that I know have the disease that are in maybe regional areas, and they certainly often aren't getting access to the things I was, or, you know, so yeah, access, cost as a patient, and your time. I mean, if you've got a job and you're doing something like this, it's working around that for reasons.
I didn't put up the schedule events in this trial, but we tried to design it so that it was the minimal amount of times you have to come in. I think it was 4 visits during this whole study, compared to some visit trials where it's like every month you have to come in for a study visit.
Yeah, it differs. But conversely, I'm happy to do it because there's a common good here. So, yeah.
Thank you very much for the questions from the audience. In the interest of time, we might move on to our next session. Thank you, Dr. Jacob Paganj and Ms. Raya. We have a little token of appreciation. Sarah's gonna Thank you. I would like to invite Dr. Yuan Hao Yang to chair the next session, which is the MATA Research Early Career Research Seeding Grants presentation session.
Good afternoon, everyone. My name is Yuan Hao Yang, and it's my pleasure to chair this ECR Seeding Grants presentation session. So the MATA Research ECR Seeding Grants screen is designed to build capacity and provide research opportunity for early career researchers at MATA Research. The grants assist successful applicants to be competitive on a national and international level through applications to major funding bodies such as the NHMRC and ARC. 2 seeding grants valued at 30,000 each will be awarded for clinical or healthy service research project and a biomedical research project. The 5 finalists presenting today have been selected by a scientific review panel consisting of biomedical and clinical healthy services senior researchers. And the selected applicants align with at least one of the 3 MATA research strategy themes, being discovery, translation, and integration. The presentations will be judged by a panel of judges, including MATA Foundation donor Ms. Nita Parkingham. Nita worked in the clinical chemistry department at MATA Adult Hospital Brisbane for 45 years. Since retiring, she served as president of the MATA Adult Hospital Auxiliary, raising funds for both adult and young adult hospitals, and was also a member of the Orthodox. Secondly, our MATA Foundation donor, Ms. Liz Coughlin. Liz is a career public servant with extensive experience in managing healthy research grants and has played a key role in establishing Cancer Australia. She now oversees Nordic local grants at the National Indigenous Australian Agency after her husband Mark passed away from a rare lymphoma. Liz donated to MATA Foundation to support Professor Mark Gandy's research on EBV positive lymphoma. And Mr. David Young, a medical consumer advocate and educator. David has been active in cancer advocacy since his lymphoma diagnosis in 2012. He works with agencies on policy and patients' guidelines, serves on various advisory panels, and has set up cancer support groups in Northern Rivers, New South Wales. He's also part of MATA Research Healthy Consumer Network. And finally, Ms. Aislinn Wood. Aislinn is currently a year 12 student at All Hallows School and is a passionate and outstanding student in their STEM program. She's joined today by fellow students and All Hallows Dean of Teaching and Learning, Mr. Scott Adamson. And the judges will collectively decide the awardees of the 2 grants during a judges' deliberation session chaired by our Director of Biomedical Research, Professor Alison Pettit. We will now have 2 clinical research presentations first, followed by 3 biomedical research presentations. Our first speaker is Dr. Hamid Karimi Rozbahani. So Dr. Karimi Rozbahani has completed 2 postdoctoral fellowships, including one at the University of Cambridge. He's currently a researcher and biomedical engineer at MATA. His work lies at the interaction of clinical and computational neuroscience, combining methods such as machine learning. His successful application of engineering techniques to neuroscience has earned him several international awards, including a Newton International Fellowship. The title of his presentation is Towards a Machine Learning-Based Tool for Localizing Epilepsy in the Brain. Welcome.
Thank you for the invitation and giving me the opportunity to present my work. So my work, let's jump into the problem. My work is on epilepsy, and the problem is human error. Let's start with the problem. I'm trying to give you some examples to see if you can discriminate two signals that I show you. Can you please tell me if these two signals are identical or different? Yeah, they are different. What about these two? Different. I agree. How about these two? Identical. Well done. These two? Look identical to me, but they are not. In just one sample, they are different. What about these two? Tricky. The lower signal is shifted upward just a bit. What about these two? The lower signal is 10% higher in amplitude. So I think I made the point of saying that we are prone to making mistakes and we might not be able to discriminate signals very well and very accurately. So this project is aimed at minimizing such human errors in epilepsy. But what is epilepsy? Epilepsy is a brain arrhythmia where brain cells send wrong signals to other parts of the brain, causing seizures. More than 150,000 patients in Australia suffer from epilepsy, which means 1 in 200 people. Medications failed in 30% of these patients. And surgery is their best chance to become seizure-free. For that purpose, we need to find out which brain areas are causing seizures and to treat them. People use, and clinicians use EEG or electrical brain activity to localize where the seizures start. And these are electrical brain activity that we collect from brain and from the scalp. We also use MRI to look at the structure of the brain, as well as nuclear medicine. But sometimes they fail. You cannot find the location of the seizures. In that situation, we use a stereo EEG and collect data from inside the brain using electrodes implanted inside the brain for 1 or 2 weeks to collect data to see where the seizures start. Still, 30% of these patients cannot become seizure-free. And the main problem is what you just saw. We are not able to discriminate patterns of brain activity from one another. Let me show you an example again. So we know that epileptic patterns are very rare, complex, and different across patients, which makes them hard to discriminate. So electrodes are implanted into the brain, signals are collected. You're looking at the signals coming from different contacts from these electrodes. And the task is to tell if these signals coming from this side of the brain, whereas these activities coming from this side of the brain, which one are causing the seizures, which one is epileptic and which one is not. And clinicians have to look at a lot of these data. I'm just showing you here 40 seconds of data, and you can see how complex it is to discriminate these patterns of activity. Clinicians need to look at more than 7,000 minutes of such data to find those patterns of epilepsy. We are not very good at it. We might miss. So we try to use machines to help us. This project is aimed at using machines. So for that purpose, we need to train a machine on seizure activity and healthy brain activity. And then test the machine to tell us whether this pattern of brain activity is a seizure activity or a healthy activity. So we have epileptic signals and healthy signals. To feed them to the machine, I'm just showing you that for every signal, we will have a point in a 2D space, which you can see here. So for every signal I'm showing you, there will be one point here. For another seizure signal, we have another point here, another point, one more point, one more point, and one more. And for healthy signals, we'll have other points. So for every healthy signal, we have other points. And then what we do is to ask the machine to find the line which can discriminate these two patterns, seizure patterns or epileptic patterns and healthy patterns. The machine finds that line and labels the right side of the line as epilepsy and the left side as healthy. And then we ask the machine, give the machine new data, which the machine hasn't seen. And if the signal, if the dot falls on the correct side of the line, it means that the machine has picked it up and discriminated that, found that it is epileptic or healthy. So for this test epileptic signal, the dot fell on the correct side. For this healthy signal on the left side. On the right side again, on the left side. On the right side, left side, and incorrectly on the left side. So in this case, the machine could successfully discriminate these two patterns of signals in 7 out of 8 instances. So with 88% accuracy. We use this technique to discriminate the signals in the brain. We have applied this method to an American data set, used machine learning, and could accurately discriminate brain signals with above 80% accuracy for epileptic patients. And we have published the results in a paper. And it is now ready, this method, to be used on other data sets, especially at MATA, where we have a big and large data set collected since 2015. So in conclusion, SEEG, or implantations of electrodes inside the brain, is growing in Australia as a method to treat drug-resistant epileptic patients. We have now developed a machine learning-based algorithm which can discriminate the signals for us relatively accurately. And if successful in this grant application, we'll be able to process the large data set which has been collected in MATA since 2015, more than 500,000 minutes of data. And our methods can benefit hundreds of patients in Queensland alone. And increasing seizure freedom by 5 to 10% only can save Australia more than $8 billion. And we plan to share our method with other Australian hospitals to improve epilepsy care in Australia. With that, I'd like to thank you for listening and to the incredible donors and supporters and their generosity towards MATA. And I'm happy to take any questions you might have.
Thank you, Hamid. Nice talk and very interesting project. So any questions from the audience? I can start with one. As I'm interested how accurate your algorithm can identify epilepsy if the patients have taken some medications that can influence the brain signal, or the patients have suffered from other similar symptoms but not epilepsy, like migraine.
That's a good question. So definitely medications, epileptic patients take medications to suppress their seizures. It is ideal to see the seizures in signals to be able to discriminate them, because seizure activity gives you some relatively reproducible effects across patients, but it is not reliable necessarily across patients. So it is ideal to see seizures to be able to discriminate them more accurately. But without seizures, I showed you the results. I showed you 2 panels of data. One is without seizure and one is with seizures when you use seizure signals. Without seizures also, you can pick up those signals. So we are planning to apply this method to the whole data set that we collect in 2 weeks of data, because not every patient has seizures when they are in the hospital. So it's ideal to to be able to do that. And our method, the preliminary results showed that it is successfully, you can successfully pick that up and find the seizure patterns, find the epileptic patterns.
Thank you. Ali, we also have some further questions.
Yep. So it had good prediction. So 80% of the cases it got it correct. But do you know what contributed to when it didn't get it correct, and how are you going to continue to educate the machine to increase your diagnostic accuracy?
Yes, exactly. So we definitely need to improve the machine. It was just the initial step that we did to show how feasible it is. The method is working and we need to improve that. And we know what directions to take. So these results that I showed you came out of more than 30 different measures that we extracted from brain signals, and we know which ones. For example, in this case, the variability of the signals can give you a good indication, a good biomarker for localization and discrimination of signal patterns from one another. So yeah, we know what directions to take, and we have methods to do that. Yes.
I think we also have another 2 questions, maybe quick questions.
Thanks, great presentation. Big question is, are you considering expanding this to the surface electrode use? Because of course, globally, a lot more data and a lot more access to surface electrodes, whereas intracerebral electrodes is for rich countries, really.
Yes, you're right. It is ideal to get out of the brain and do everything from outside. The problem is that the surface electrodes do not give you very enough localization power. Otherwise, we wouldn't have gone inside. So we are working on that aspect as well. Hopefully, we can improve the methods in a way that we can get good accuracy. We are not there yet, but we are going in that direction.
So we have the last question from Fahad.
Thank you. That was a great talk, Hamid. My question is simply, once you have identified a person through your machine learning, I'm presuming they could then go straight on to some form of treatment. What would that treatment be?
Yeah, I try to not mention that because of the nature of the session. So it is mostly resection of the brain areas safely if they can be resected. So the first step is to localize where the seizures and the epileptic patterns start, to either resect them through different methods. So either resected, laser ablation, or some other forms of treatment which can keep the tissue inside the brain.
Thank you, Hamid. So now we move to our second speaker, Ms. Bronnie McAllenden. Bronnie is an advanced practice physiotherapist with 17 years clinical experience in neonatal and pediatric intensive care at Mater Hospital. She was awarded a master's degree in respiratory physiotherapy for ventilated infants in 2019 and is now researching new methods to improve respiratory care for preterm infants. Her current research project explores lung ultrasound to improve efficacy, safety, and individualization of respiratory physiotherapy in preterm infants. The title of her presentation is Measuring Chest Physiotherapy in Premature Babies Using Lung Ultrasound.
Thank you. Hello. Thank you for the opportunity to speak to you this afternoon. As you heard, my name's Brona, and I'm an advanced practice physiotherapist working just down the road at the Mother's Hospital Neonatal Critical Care Unit. And I'm going to speak to you this afternoon about an area I'm really interested in, which is measuring chest physiotherapy in premature babies using lung ultrasound. So in premature babies, chest physiotherapy refers to a combination of specific techniques, including positioning, cupping or tapping over a baby's chest, and airway suctioning. And these techniques are regarded as an important part of care for many premature babies, particularly those needing a ventilator to breathe, because they can help the baby clear the airways and make it easier for them to breathe, particularly when a baby is either too sick or too immature to do this by themselves. So premature babies, or those babies born before 37 weeks gestation, are at high risk of lung problems, which can lead to many of these babies needing long-term oxygen therapy and having recurrent breathing problems and chest infections in childhood. And chest physiotherapy can help premature babies recover from certain lung problems, for example, lung collapse, which is where there is a buildup of secretions in the small airways, making it difficult for the baby to breathe. It's important to note, however, that chest physiotherapy can be of no or little benefit to other types of lung disease. And so it's really important for a neonatal physiotherapist to be able to accurately diagnose what lung disease is actually occurring so that we can safely and effectively apply chest physiotherapy. And at the moment, chest physiotherapy diagnostic tools are quite minimal, and a lot of them have limitations. So it becomes very difficult for us to differentiate what is going on in the lung. And so a new technique for physiotherapists, one not traditionally used, called lung ultrasound, has been suggested as a new, more accurate and safer way for us to be able to see what's happening in the lungs and guide clinical decision-making. So we know from the literature that lung ultrasound can give us more accurate information than we can get from a stethoscope. It can give us similar information to what we can see on a chest X-ray, but without putting a baby through radiation. And it's just as good at diagnosing certain lung problems as a CT scan of the chest, which is currently the gold standard for lung imaging. Unlike CT, though, lung ultrasound can be done very, very quickly and without moving the baby from their cot. And this makes lung ultrasound a much faster and safer way of imaging a premature baby's lungs without having to move them to a different part of the hospital and risking deterioration. A bedside lung ultrasound, for example, might take a minute or a couple of minutes compared to a CT, which could take well over an hour. And as well as giving more detail about what's happening in the lungs, lung ultrasound also allows the physio to see very specifically what's happening in different parts of the lungs. So it allows us to basically treat very, very specifically and in a targeted manner when we see a disease process that we can assist. So to investigate whether chest physiotherapy delivered lung ultrasound can be safely, accurately, and beneficially used with premature babies, we have developed a 5-phase research program and a successful seeding grant will enable the phase 4 study to occur. And it looks a little bit like this. So basically, the primary objective of the study is to measure changes in the degree and location of lung collapse using lung ultrasound to see what effects chest physiotherapy has on improving lung collapse in premature babies. And we hope to recruit 10 premature babies from the Mater Neonatal Critical Care Unit who have a breathing tube and who have changes on their X-ray that are reflective of lung collapse. Babies will then undergo a physiotherapy assessment, including with a lung ultrasound, and an individualised targeted treatment plan will be established based on those results and delivered to the baby. The babies will then undergo another lung ultrasound immediately after chest physiotherapy and again 24 hours later in order to measure any immediate and longer term effects from chest physiotherapy. And we suggest that we will be able to measure changes that we can cause as a result of chest physiotherapy much better using lung ultrasound than with the current tools that we have. So the anticipated benefits of this research are many, but we think the main one is that results will help physiotherapists determine which premature babies are likely to respond the best to chest physiotherapy, and improvements hopefully that we can make in the baby's lung function in the early stage of their treatment will hopefully translate to better outcomes in their long-term lung health. And we see this study as really important because there's very little clinical evidence out there looking at chest physiotherapy in premature infants, with very few studies being published in the last 20 years, which means that a lot of what is known and what is done is based on adult studies, but premature infants are not just small adults, and their lung disease and their lung development is very different and very unique to them. And so because of the lack of current research, chest physiotherapy is still very largely underutilised in neonatal units around Australia, and premature babies are potentially missing out on an important lung treatment. So we hope that through doing this study, we can try and identify whether we can have a similar measurement improvement using lung ultrasound as has been found in adults. We hope to be able to establish some practice guidelines and also some education programs to work toward hopefully getting this into routine practice. We hope to do that in correlation with universities and MARTA education. And also as a part of the MARTA neonatal critical care research program, we hope not only to improve clinical chest physiotherapy practice, but also improve outcomes for our tiniest and fragile patients. Thank you. And particular thank you to our donors and supporters. Without you, we could not do what we do. So thank you.
Thank you, Bruna. We have time for one quick question. Right.
Is this being done concurrently with physiotherapy, or is it in between sessions?
So the idea we use it, hopefully we will work to using it as an extra and a better assessment tool than what we currently have. So we'll use it to identify what we think is going on, treat the baby according to that, and then use it again to see whether we have made the improvements that we hope we have.
I think we got one more question.
Rona, you described a potentially really valuable study design that I think, if I understood it correctly, involves using each baby as their own control. So doing a before and after measurements, and then a longitudinal measurement 24 hours later. But have you considered doing a no intervention control as well for this study? Would that improve the the level of evidence that you derive?
Yeah, thank you, Helen. I mean, that's a really good suggestion. We are kind of starting to get this trial underway and starting to put the pieces together. So yeah, thank you for that suggestion.
Thank you. Now we move to the biomedical research presentations. Our next speaker is Dr. Charles Bell. Dr. Bell completed his undergraduate degree in genetics and evolutionary biology at UQ. He began his research career at MARTA Research, performing his honors, where she was awarded the University Medal. Dr. Bell then pursued a PhD with Professor Mark Dawson in Melbourne, leading two major projects published in Nature Communications and Nature Genetics. He has recently returned to Queensland to be closer to his family and aims to start his own research group in the coming years. The title of his presentation is Conducting the Orchestra, New Approaches for Drugging the Undruggable in Development and Disease.
Thank you for pre-selecting me for the ECR thing, and I'm very happy to be presenting today. So I recently had my first child, and when my wife announced to me that she was pregnant, she gave me a piece of paper with a tiny black dot drawn in the middle of it. At first, I was very confused until she said, "Congratulations, we've made a tiny clump of cells the size of this dot." When I look at my 1.5-year-old daughter now, walking, talking, and dancing to the Wiggles, I think back at that moment and how miraculous it is that that tiny clump of cells could turn into this human being using just the genetic instructions present in those cells. How does this happen? How is the miracle of human life possible? Well, some of the first important clues emerged in the 1960s when Professor John Gurdon won the Nobel Prize studying frogs of all things. He first showed that the cells in the frog's intestine contain the same genetic material as the cells in the frog's egg. He could take the genes present in the intestinal cell and put them into the egg, and this could produce an entire frog. This was very important because it revealed that the genes present in the intestinal cell are the same genes present in the frog's egg. And to form intestines, a brain, a lung, or any other tissue doesn't require changes in the genetic material. Instead, the cells become different from each other by turning on different sets of genes. In other words, it's not so much the genes in our genome, but actually how they're controlled that enables all these different cell types and the whole human being to develop. But how does this actually work? How does that tiny clump of cells become a walking, talking baby? In order to turn the right genes on at the right time, we have special genes in our genome called transcription factors, and they act to orchestrate the entire process. These genes have the unique ability to directly read the DNA sequence and turn on the right genes at the right time. If all goes well, we get a healthy baby. But if things go wrong, then it can result in developmental defects, cancer, and other diseases. While the general idea behind this process has been understood for a long time, we have struggled to really understand how these transcription factors work and how they manage to turn on and off the genes that they control. I've spent the last 6 years trying to address this fundamental question. The results of this work have recently been published in one of the most prestigious scientific journals, Nature Genetics, and I think has provided some key insights into this problem. So to help summarize what I discovered, I can use a simple analogy. The cell is a lot like an orchestra. There are a lot of different genes, like there are a lot of different instruments. And we want the genes to be switched on at the right time, just like we want the instruments to play at the right time. If everything works well, then we end up with a beautiful song. Transcription factors are like the conductors. They coordinate the entire process and make sure all the instruments play at the right time. I'm interested in how these conductors can do what they do, how do they control the entire orchestra, and how do they get everyone to work together to make that song. To study this, I examined 9 of these different conductors or transcription factors in great detail and compared and contrasted how they were able to control the orchestra. To do this, I isolated each of these individual transcription factors and used a revolutionary technology called CRISPR to identify how they work. The results and data are very complicated, as you can probably tell from this plot, and they have a lot of different implications. But the biggest take-home message was that each of these conductors had their own special approach. Each of them worked differently, which shows that there are many different ways to control the orchestra and potentially many different ways for us to help control this process in development or disease. So you might be thinking this is all very interesting, but why does it matter and why should we care? Well, just as a good conductor can get the orchestra to play beautiful music, if the conductor goes mad, they can start to produce horrible music. In much the same way, if transcription factors activate the wrong genes at the wrong time, this can result in diseases such as cancer or problems with development. In the future, I don't want to just understand how these transcription factors work, but I also would like to manipulate their activity in order to help cure disease. Drugging transcription factors has been a long-standing challenge. so much so that these genes have often been called undruggable. But clearly, since they play such a fundamental role in regulating how cells behave, drugging them would be a very promising strategy. Recent technological advances, such as mRNA technologies, which is similar to the technology used to develop the COVID vaccine, have provided a new class of drugs that have opened up this problem. The issue is we don't know which part of the transcription factor to target. If we target any of these, some of them may not work. My project will attempt to map the regions of these transcription factors that are most promising to target, and in collaboration with the base mRNA facility at UQ, develop new drugs to target this class of proteins that have long been considered undruggable. Ultimately, over the coming years, my hope is that by understanding how transcription factors function, we can develop new therapeutic approaches to prevent them from driving disease and ultimately become the conductors of our own orchestra. Thank you so much for listening, and special thank you to the Mater Foundation for supporting us here at Mater.
Nice talk. Questions from the audience?
Thank you. That's a very exciting project and wonderful work. And when you say we don't know which bits to target, there must be some candidates, right?
Yes. There are parts of the proteins that are well understood, but they're often shared across multiple different transcription factors, so you won't achieve specificity. The parts that we want to target are quite divergent between transcription factors. The issue is the regions which are divergent are also poorly understood, partly because of their divergence. So in order to target them, we need to better understand how those parts of the protein are working. And that's what I'll be focused on.
So just when thinking about drug use, do the transcription factors work the same way with every single gene that they control? And do they continue to work in the same way across different developmental stages? Or is it going to be a different fix for different things?
Yeah, that's a really good question. They probably won't, as we know, biology is quite messy. But it's just another tool that we can use to be able to manipulate the system. So a lot of the therapies that have been developed in the past don't have much specificity. Things like chemotherapy, they just target division. So this would be a lot more specific than something like chemotherapy, but obviously there's always the potential for off targets in different cell types. So if the technology develops to a point where you can start to target these things to specific cell types, then it will be very powerful. But at the moment, we don't really have those technologies available.
Thanks for the excellent talk. It's really exciting work. Can I just ask you from a clinical perspective, do you have a model of disease that you would look at, I guess, examining first?
Haven't got that far yet. But yeah, obviously, some of the transcription factors I've studied in my previous project are key regulators of cancer, like P53, key regulators of inflammation, like NF-kappa B. So any disease in which those things are implicated could be a potential model. When we go to start testing some of these drugs that we develop, then we'll have to form collaborations with some people who are working on diseases where these transcription factors are important. But at the moment, I haven't thought that far ahead.
Thanks, Charles. Our next speaker is Dr. Raphael Ritchie. Dr. Ritchie completed his PhD in neurosciences at the University of Tasmania in 2021. He did postdoctoral training at the Queensland Brain Institute and more recently joined Mater Research. Dr. Ritchie is an early career neuroscientist investigating the capacity of glial cells and myelin to shape neural circuits. His research aims to understand how brain function is regulated during development and in healthy aging, and the pathological signals that enable neurological disorders such as autism and multiple sclerosis. The title of his presentation is Investigating How COVID-19 Impacts the Multiple Sclerosis Brain.
Thank you very much all for the opportunity to be here and sharing this really interesting project that I've been working so far here at MATA. So as you know, the brain is this amazing organ that allows us to actually walk, talk, think, feel, and behave as we do. And this is all because of specialized cells that I have in yellow up there called neurons. So those cells talk to each other via electrical signals, just like in a relay race, or like wires. And just like wires, they need insulation, which is done by a substance called myelin that I have in green up there. So in MS, or multiple sclerosis, the immune system ends up attacking this myelin and this insulation, causing this signal transduction to slow down and resulting in the devastating symptoms that people with MS experience, like losing the ability to walk, talk, and having very strong fatigue and all of that. Over 2.8 million people worldwide are living with MS right now, and more than 33,000 in Australia alone. And every single day, 1 or 2 Australians are diagnosed with MS. Now, COVID-19 is the coronavirus disease that I think is not news for anyone anymore in this room, and it resulted in the pandemics in 2020. But now it's becoming more endemic, just like flu, and more people are getting infected and reinfected, and new strains are coming out. It has common symptoms just like flu, but some people may experience more severe symptoms like extreme fatigue, memory and concentration, loss of memory and concentration, trouble focusing, and changes in behavior, and even inflammation in the brain. And those are key symptoms of something happening in the brain. And that's really important because then it raises the question, what is COVID-19 doing the brain of someone that has already a neurological condition like MS? So has already some of those symptoms. And interestingly, reports from MS Australia from 2017 and 2021 showed an increase of 27% of MS cases. And it makes me wonder, what is the contribution of COVID-19 to this increase as it happened in between those 2 time points? Now, COVID-19 invades the brain via 2 different pathways, so 2 different ways. So the first one is via the nose, so it's an airborne virus, right? So we breathe it in, and because we have neurons that allow us to smell, sense smell, it uses that as a gateway to actually invade the brain. But COVID-19 also causes a systemic inflammation, and by doing that, it uses immune cells that are infected to actually invade the brain, and just like a Trojan horse ends up in there. Now, studying both COVID and MS, it's really challenging because of the challenging models, preclinical models that we have to use, and the very different expertise that we need to have to actually study those 2 diseases. But here at MATA Research, we have the perfect environment to perform these studies. So I was able to establish and have now an ongoing collaboration between a team of neuroscientists, including myself, and immunologists, where we can associate multiple sclerosis research and COVID-19 research together in one model, in one place, and study the association of both diseases. I'll then use microscopy to look at the cells that insulate the brain, like I mentioned before. Those ones are the ones that are lost in MS. And I'll look at these myelin fibers and also the cells that actually produce this brain insulation. And with that, I'll be able to determine the extent of cellular damage caused by COVID in the context of MS, and also detect possible molecular and cellular targets for brain protection. We all know cholesterol, and we know that the good cholesterol is really important for brain function. And 70 and 80% of this cholesterol is present in myelin, which is responsible for brain insulation. Now, during normal metabolism, this cholesterol is converted in what we call oxysterol, or oxygenated forms of cholesterol. And those are really important molecules for proper brain function and also immune system function. So when you have too much of them or too little of them, it can be detrimental. Now, with this project, I'll be able to investigate how both COVID-19 infection and MS is actually altering the oxysterols and those signals in the brain, and whether or not we could use them as a potential target for brain protection after COVID-19 infection. And with this project, then we'll be able to inform both clinicians and the population in general how COVID is actually affecting the brain, and understand how it is more specifically affecting brain insulation, which is kind of critical for people with MS that has already an impairment in brain insulation. And it could be also really important for translating it to other neurological disorders that share common characteristics with MS. I will then investigate whether oxysterols can protect the brain against COVID-19, and especially in the context of MS. And we could potentially use those as pharmaceutical targets to actually treat the disease or treat the effects of that this could be causing in the brain. And more importantly, we will be able to then prevent unexpected brain effects in real time. So what is happening right now is we don't know what is going on in the brain with COVID infection, but then people are being infected and reinfected. So we want to have this opportunity to do a jump start and understand this before and find ways to prevent any issue that could arise from that. Thank you very much for the opportunity, and a special thank you for my supervisors and collaborators, and of course for our donors that make all this work possible. Thank you.
Thank you, Raphael. We have time for one quick question.
Hello, thank you for the talk. I have a question. How at the beginning you mentioned that the incidence of the multiple sclerosis increased and that COVID is responsible for it. How you can tell that it's actually COVID?
No, it's not responsible. It just makes us wonder the contribution of COVID to that. So there are multiple factors that could contribute to an increased incidence of MS. And we know that MS is on the rise. And things like sun exposure, vitamin D, and lifestyle could be affecting it, including viral infection. So viral infection is a big thing. And that's why with this project, we wonder what is the contribution of MS with that, the contribution of COVID to that increase.
Thanks, Raphael. We now move to our last speaker, Dr. Stephanie Walkman. Dr. Walkman completed her PhD in 2022 at the University of Otago, New Zealand. Shortly after, she began her career as a postdoctoral researcher here at MATA. Dr. Walkman's research builds on her passion for understanding the complexity of the female reproductive system, where she used genetic approaches to investigate how the ovarian supports the growth of eggs. The title of her presentation is Measuring Jumping Genes in the Egg.
Thank you, Yuan Hao, and thank you everyone for the opportunity to present to you all today my work on jumping genes inside our egg cells. After failing to conceive for several years, Julia and Alice each turned to medicine for help in building their families. And they're not alone, with 1 in 6 Australian couples experiencing the isolating and often invisible struggle of infertility. Julia and Alice will spend months going through the enormous physical, financial, and emotional tolls of fertility treatments. And at the end of it all, Julia can share the happy news that she is going to have a child. Unfortunately, Alice cannot say the same, and no one can tell her why. The reason current approaches to fertility care work for some of us and not others isn't clear to scientists or clinicians. And perhaps the largest mystery of the female reproductive system is actually the egg cell itself. Because we only produce one mature egg each cycle, one of the first steps of fertility care is to use hormonal treatment to tell our ovaries to start producing lots of eggs at once and to do it fast. And for many women, like Julia, hormonal stimulation is a critical step towards her successful pregnancy. However, new research has shown us that for other women like Alice, this hormonal stimulation might actually reduce the chances of her success. But what is the underlying reason for this difference? Long before meeting with sperm, an egg begins to carefully organize its genetic code or its DNA sequence as it grows. And that maturing egg keeps all of the important molecular signals and messages in a tightly controlled order, so that when the time comes, the embryo has everything that it needs to kickstart development. The new research has shown us that when we stimulate eggs to mature quickly, like when we have hormone treatment, some egg cells aren't able to organize their DNA effectively. And that disorganization then has a flow-on effect to the embryo, reducing the chances of successful implantation and pregnancy. And we think that one particular part of our DNA is taking advantage of this disorganization for its own gain and to the detriment of our egg's health. And that element is called a jumping gene. Jumping genes are selfish parts of our DNA that make up about 17% of our genetic code. And their goal is to survive by shifting from one generation to the next. And they do this by literally jumping throughout our DNA, copying in one location and pasting into another, inside our eggs, our sperm, and our embryos. Normally, our egg's DNA is really good at stopping jumping genes because they've organized it in such a way that sends the jumping gene a stop signal. We think that after hormonal stimulation, some eggs haven't been able to properly establish those jumping gene stop signals, allowing them to become active. And active jumping genes can harm our cells by causing inflammation, DNA damage, and mutation. And so we want to know if our eggs are more vulnerable to jumping gene activity following hormonal stimulation due to changes in the DNA organization. To answer this, we'll be implementing a new, extremely precise method developed in the lab of our international collaborator that I will establish here at MARTA. First, we will use a laboratory model to collect individual stimulated and unstimulated eggs across different stages of egg maturation. We will then collect the genetic material from each egg and measure the jumping gene activity so we can see at which point during the egg's maturation they're most active and if hormonal stimulation increases that activity. As for the egg's stop and go signals, we aim to use specialized sequencing approaches to see where the signals are altered in stimulated eggs so that we might better understand how the jumping genes are becoming active. Importantly, we don't just want to measure jumping gene signals as a number. We actually want to be able to see the jumping genes inside our egg cell. And to do that, we'll use a cutting-edge imaging technology, like I've shown on the right, where we can see those red jumping genes active inside that round egg cell. With my expertise in female reproduction and jumping genes and MARTA's strong clinical background in maternal health care, I'm hopeful for the future of this work and its alignment with the MARTA research mission. By understanding how our eggs respond to hormonal stimulation, we expect to discover how jumping genes might reduce the chances of successful pregnancies. In the future, we plan to use this information to test new approaches for fertility care in a laboratory model that avoid widespread changes to the egg's DNA. It's our hope that this work will generate promising and safe alternatives to current care that could one day in the future be translated into clinical trials and eventually integrated as part of standard fertility care practices. At the moment, we don't know why current clinical approaches haven't been able to help Alice. We hope that by building a strong foundation of knowledge, that someday we can develop alternative treatments that work for Alice and for thousands of women like her. I would like to thank the members of the Richardson Lab and that of our international collaborators. Most importantly, I would like to thank those of you who generously donate to Mater Foundation. Without you all, my work and the work of all our presenters today wouldn't be possible. Thank you.
Nice talk. Any questions for Stephanie? Charles?
Yeah, so I think normally that process where an egg goes from laying dormant, so not growing at all, and becoming an egg that's fully able to mature and be ovulated is quite a long process, and it's really controlled with only sort of one egg making it that far each time. So I think that when we stimulate the eggs to mature with many at one time, that could have an effect on how they organize their DNA and that rapid pace of that maturation as well. So I think that's part of the exciting part of this work, is that we can look across maturation and see when it might be having that effect.
So the fertility treatment to increase egg production is basically simulating normal biology, but just putting it on superdrive. So is this also potentially a problem why Alice was infertile in the first place, and that her own, even with the normal pace of this process, her DNA doesn't know how to protect itself properly?
Yeah, that's a really good question. So I think you're probably onto something there, and that there could be definitely an underlying role in jumping genes in general fertility issues. And I think that's something that definitely warrants further research. And I guess that's the hope from looking in our unstimulated eggs in this work, that we might be able to analyze that as well. So not just looking in the clinical aspect, but at the scientific aspect too.
I think we got our last question.
The good thing about these sort of seminars is you get lots of ideas to do another 20 years work. Ali's given you one, I'll give you another. There's a strong relationship, as I'm sure you're aware, between stimulated multiple pregnancies and deleterious outcomes for the offspring, in contrast to natural multiple births, where there is not that association. So I guess it's a follow-on from the previous two questions. It's another potential impact of your work would be to improve the outcome of stimulated multiple pregnancy offspring.
Yeah, thank you. That's true, and it's something I'm aware of, but haven't thought yet about how this might apply to that. So thank you.
Thank you, Stephanie. Please join me again to thank all the speakers and our judging panels. And I'd like to hand over to Dr. Hallie Kimball to chair the Mater Research International Travel Award announcement and presentation session. Dr. Hallie Kimball is an early career clinical psychology researcher and clinician at Catherine's House for Mothers, Babies, and Families. Her research investigates many facets of perinatal and infant mental health.
Thank you, Dr. Yang. So it is my absolute pleasure today to present the winners of the Mater Research International Travel Awards. The competitive Mater Research International Travel Awards assist towards conference expenses for 4 HDR students to present Mater research data at an international conference. These awards also provide support towards additional travel to visit and/or interview at international laboratories, in conjunction with conference attendance, to assist the student in securing a post-PhD position. 3 of these awards are valued at $3,500 each, while the David Srizier Student Training Award is valued at $6,000. So we'll now watch a 1-minute video by Dr. Lucy Burr that presents this award.
Hi. Thank you for inviting me to speak about Associate Professor David Srizier, the namesake of this premier award. I'm Dr. Lucy Burr. I'm a respiratory physician here at the Mater Hospital. So David was a respiratory physician here at Mater, and he was a true rising star, both in the clinical and in the research world. I first met him when I was a specialist respiratory trainee, again here at the Mater in 2011, 10 years ago. He was a passionate clinical supervisor and a dedicated physician, which was exemplified by his work with his cystic fibrosis patients. He was incredibly caring and taught me how to practice medicine with empathy and humility, even when having difficult end-of-life conversations with young people suffering with disease. Despite his heavy clinical load, at the time he was Director of Physician Training at the Mater, Director of Respiratory Medicine and also Chair of several committees, including in the Lung Foundation Australia and the Thoracic Society. He was also leading the first Australian study investigating the role of erythromycin, a macrolide antibiotic, in the treatment of bronchiectasis. Bronchiectasis is a severe lung disease characterised by inflammation, infection, sputum production and cough. This landmark study was published in 2013 in JAMA, a very prestigious journal, and it sprung David really into the forefront of global respiratory research. The samples collected during the BLESS study supported at least 2 PhD students, including myself, with David nominated as Principal Supervisor for both. The unstoppable David then set up MARDA's first respiratory biobank, a diverse repository of clinical samples from patients suffering from respiratory disease, which is still ongoing and currently supporting research into COVID-19 vaccines, as well as treatments for cystic fibrosis infection and lung cancer. David was also instrumental in setting up the National Australian Bronchiectasis Registry, which is currently thriving with nearly 2,000 participants 5 years on. Sadly, David passed away suddenly and tragically at the age of 45 back in 2015. He left behind 3 beautiful children, the oldest of which was only 8 at the time of his death. It has been my honour to help carry on some of his work here at the Mater, and I'm delighted that this award has been named in his memory. Congratulations to the winner this year.
So the winner of this year's award is Shivam Vora. Shivam Vora is a final year PhD student in the Smiling for Smitty Cancer Cell Cycle group at Mater Research. Shivam completed his Masters in Biotechnology at UQ in 2020 and graduated with the Dean's Commendation for Academic Excellence. After completing his master's degree, Shivam started his PhD at Mater Research under the supervision of Professor Brian Gabrielli in 2021. His work focuses on polyploidy induced by the inhibition of aurora kinase B and the genetic background that determines the outcomes of this inhibition. Congratulations.
Thank you for that introduction. And thank you so much for this award to the Mater Foundation and everyone here today. So the talk that I'm giving today is titled Creating Giant Cells, a Novel Approach for Targeting Cancer. So first, I'd like to talk about my recent travels to Europe. So the Dr. David Cerisier Student Training Award assisted me in funding my trip to the Netherlands, where I attended the European Association of Cancer Research Congress, which was held in Rotterdam. I attended several talks and connected with many fellow researchers from all over Europe and even the rest of the world, and it was a great experience. Following that, I visited the lab of Professor Suzanne Lins at University Medical Center Utrecht. She's an expert in the field, and I presented my findings to her lab. And today I will be presenting some of the work that I have presented at these meetings. So my PhD work has focused on cancers that have specific mutations in the 2 main anti-cancer genes, P53 and RB. These are also known as tumor suppressor genes. These include nearly all cervical cancers and small cell lung cancers. And lung cancer's impact on society cannot be understated. There were 15,000 cases of lung cancer diagnosed in Australia last year, of which about 15% represent small cell lung cancer. Additionally, lung cancer is the leading cause of cancer-related death in Australia, and small cell lung cancer specifically has an extremely low 5-year survival rate of about 3%. Additionally, radiation and chemotherapy that are used in the treatments of these cancers have severe side effects that can have long-lasting impacts on the patient's life. So the question is, how can we treat cancer more effectively? And we always think of chemotherapy, and chemotherapy has greatly, has done a lot for the treatment of cancer. But we can, everyone knows that it comes with a whole range of adverse effects. This is because the chemotherapy is not completely effective at discriminating between the normal cells of the body versus the cancer cells. Therefore, there is a need for the development of new targeted therapies that are better able to distinguish between the two and selectively kill just the cancer cells. So the targeted therapy that I'm working on is a drug that targets a protein known as aurora kinase B that is essential for cell division. These are some movies. So first we will look at the control cells, a normally dividing cell that enters cell division, and we see them exiting as two identical daughter cells, exactly as it should. Whereas with the inhibition of aurora kinase B, we can see that the cells attempt to divide, but there are no resulting daughter cells, only the same initial cell with twice the amount of content. So to elaborate a bit further, this is a representation of what normal cell division looks like. It's a very controlled and timely process, and we can see that with the passing of time, there is a huge increase in the number of daughter cells. With the inhibition of the drug, the cells are not able to divide, and the inhibitor that we use is actually, aurora B inhibitors are very efficient at preventing this resulting cell division. So with the normal cells of the body, the cells, they would fail cytokinesis, or the exit from division, and then they would arrest, meaning that they would stop attempting to divide. Whereas with cancers that have the mutations in the tumor suppressor genes that I mentioned, which is P53 and RB, these cells don't know when to stop, and they continue to divide or attempting to divide and fail, accumulating more and more content and becoming what we call giant cells. In this particular case, these cells are known as endomitotic polyploid giant cells, or EPGCs for short. So this image, is an image from fluorescent microscopy, and we can really appreciate the difference in the size of these cells. So both of these images are of the exact same cells taken at the same magnification, the only difference being the treatment. After 6 days of treatment, we can really see there's a huge increase in the size of these cells. And we hypothesize that cells that are so far removed from their biological state must harbor some vulnerabilities that we could exploit by using another therapy in combination. So to pursue that, we conducted a drug screen last year, from which we got, so that drug screen, which examined a very large number of compounds to see if we could selectively kill the giant cells, or the EPGCs as we call them. And what we got was 30 promising hits, and this is a representation of one of them. So here we can see that with the increasing concentration of that compound, the green line here, which represents the giant cells, there's a systematic increase in the cell death in these cells, whereas the normal cells of the body are relatively unaffected. And my work continues to investigate other vulnerabilities, particularly metabolic vulnerabilities in these giant cells, and see if that could be exploited in targeting these and selectively killing these giant cells. And finally, I would like to thank, firstly, the incredible donors and supporters for their generosity towards Mater. And I would also like to thank everyone at the lab, and particularly my supervisor, Professor Brian Gabrielli. Thank you.
Thank you, Shvam. So the next winner of the Mater Research International Travel Award is Caitlin Flynn. Caitlin Flynn is a PhD student in the musculoskeletal genomics group at Mater Research. She applies statistical genetic methods with the aim of identifying new drug targets to treat osteoporosis and prevent fractures. Congratulations.
Okay, hello and good afternoon, everyone, and thank you so much for joining. It's a privilege to be standing here as one of the award winners for this International Travel Award. So this award helped fund my trip to Europe earlier this year, and you can see some of the fun pictures attached on the screen. So I flew to Marseille in France, and I was able to attend the European Calcified Tissue Society's annual meeting, and this is actually the largest bone-related conference in Europe. And then after that, I flew over to the UK, where I visited at the University of Bristol and attended 2 lab visits, one at the Hammond Lab and then at the Integrative Epidemiology Unit. And what was really great about this trip was I was able to meet collaborators that I've been working with over Zoom for the past 3 years, but also make some new collaborators that I can work with in my PhD as well as after my PhD, which is awesome. So as mentioned, I work in bone fracture and preventing bone fracture. So I thought I'd start with some things about bone fracture that maybe you don't know. So in Australia, there are 500 bone fractures a day. So that's double the amount of people that could fit in this auditorium right now. But I'm not just talking about the fractures from children falling out of trees, I'm talking about fragility fractures. So these are fractures that happen when you fall from a standing height. So normally this happens in people of an older age or people with diseases that lead to weakened bone, such as osteoporosis. And while this might not sound too serious, what is serious is that 25% of people with a hip fragility fracture will die within 6 months of their injury. And 200 million people around the world have osteoporosis. So it affects both men and women, with 1 in 3 women over the age of 50 experiencing an osteoporotic fracture and 1 in 4 men. So identifying how our bones get weaker as we get older is important in trying to identify new drug targets to better prevent bone loss and promote bone growth in affected individuals. So inside our bones, we have cells that are responsible for building new bone and cells that are responsible for removing old and damaged bone. And these cells all work together to make sure our bones stay strong and healthy. But as we get older, this gets knocked out of sync and our bones begin to get weaker. So my aim in my research is to identify which of these cells are involved in maintaining our bone strength and which genes are responsible for controlling these cells, because genes actually make an ideal drug target. And this is because if we can target the genes that are only expressed in our bone cells, we can prevent the drugs from affecting other tissues and organs in our body. So what did I do? So I did a genetic study of 600,000 people and identified 1,600 genes that were associated to maintaining bone strength. We also narrowed down these genes to being highly expressed in 4 specific bone-related cell types. But as you can appreciate, 1,600 genes is still a very big list to investigate to find some drug targets. So what we then did is we looked at associations of these genes with fracture. So do any of these genes increase your risk of fracture, or do any of these genes decrease your risk of fracture? And excitingly, we were able to narrow down this list to only a handful of genes that we wanted to look at. So what happens now is I take that handful of genes and I work with some of those amazing collaborators that I've just met overseas, and we can work on implementing some functional studies and some preclinical studies to determine the effect of these genes on the bone specifically, as well as the safety and efficacy of targeting these genes with drugs to treat weak bones. So with that, I would just, oh, never mind. With that, I'd just like to thank the Mater Foundation and all of the donors who aided in facilitating this award. Your generosity is fundamental not only to our research, but also to the dissemination of our research around the world, which helps us find people to work together towards the treatments. So thank you.
Thank you, Caitlin. Our next winner of the Mater Research International Travel Award is Sripana Bomik. Sripana is a third-year PhD student in the Pregnancy and Developmental Lab at Mater Research. She is currently attending the IFPA conference in Canada, lucky, and has pre-recorded a presentation for us. Her research focuses on how stress, anxiety, and depression affect pregnancy by studying the body's stress receptors. Her work aims to shed light on how these emotional and psychological factors can influence pregnancy outcomes.
Hello everyone, I'm Sriparna and I'm going to be talking about the effect of stress and inflammation on glucocorticoid receptor isoform expression. The Mater International Travel Award has very kindly funded my trip to the International Federation of Placenta Association 2024 meeting. This is being held in Montreal, Canada this month, and I'm going to be taking you through some of the data that I'm going to be presenting at this conference. So my PhD focuses on the placenta. Specifically, I look at glucocorticoid receptors in the placenta and how they contribute to placental complications and pregnancy complications like prenatal stress, anxiety, and depression. Glucocorticoid receptors are stress receptors and they're present throughout our body. During pregnancy, they're also present in the placenta. And these receptors can be activated by our stress hormone or cortisol. And once they're activated, they can exert their anti-inflammatory effects, which means that they can prevent inflammation. However, in some pregnancy complications like stress, anxiety, and depression, there is an increase in the stress hormone, but at the same time, there's an increase in placental inflammation as well, which is contradictory because we would expect cortisol to reduce or prevent inflammation. But it doesn't, and we don't know why or how. We think that the answer lies in one of these receptor isoforms, in the sense that some of these receptor isoforms could be pro-inflammatory instead of being anti-inflammatory. And my goal is to find out which receptor isoform mediates increased inflammation in the presence of increased cortisol. So here's what we found. We found that in 2 different Australian cohorts, in women who had moderate to extremely severe stress, anxiety, or depression, or SAD, these women had an increase in this placental glucocorticoid receptor isoform called GR-alpha D1. And this was the only isoform that was altered. It was also associated with an increase in pro-inflammatory cytokines in the placenta and a decrease in anti-inflammatory cytokines. So naturally, we wanted to find out what the role of this isoform is. And we've used a placental explant model to figure it out. This basically means that we've collected placentas from cesarean sections. We've chopped it up and we've cultured them in dishes like this with culture media. And then I've treated these explants with an inflammatory mediator in the presence or absence of cortisol. And I've measured the glucocorticoid receptor isoform expression. And I've also measured inflammatory marker expression. Here's what I found, and here's what I'll be presenting at the conference. I found that in the presence of inflammation and in the presence of inflammation and cortisol, GR-alpha D1 is increased compared to the controls. And this has been associated with an increase in all of these pro-inflammatory markers. So to answer the question that we originally asked, how does high cortisol and high inflammation coexist? We think that it is because of this GR isoform, GR-alpha D1. And what this does is that it is increased in the presence of inflammation, but it can further promote placental inflammation and inflammation in the mum. And that can not only negatively affect stress, anxiety, and depression, but it can also negatively affect overall maternal and fetal health. So with this, I would like to thank my supervisors, my team members, the Queensland family cohort, and everyone, all of the women who have donated their placenta for our study. I would like to thank the ARC Discovery Grant for funding my lab work. And lastly, a massive thank you to all the incredible donors and supporters for their generosity towards Mater, to all the people who've left gifts in their will to Mater, and a thank you to all of our trust and foundation partners. Thank you, and thank you for listening.
Our final winner of the Mater Research International Travel Award is Tanya Marsden. Tanya is a PhD student with the Stillbirth Centre of Research Excellence. Her study is looking to improve the approach to stillbirth investigations by examining the utility and quality of the investigations. Tanya is a principal scientist in anatomical pathology with 24 years of experience.
Thank you, everyone. I would like to begin today by acknowledging the traditional owners of the land on which we gather and pay my respects to elders past, present and emerging, and extend my respects to any elders present here or Aboriginal people present here today. My presentation is actually on stillbirths, and I'm very mindful that this is a very sensitive topic, and I would like to acknowledge all women and families who have experienced a stillbirth. I'm very mindful of this, and so I will actually be using an analogy of a chocolate cake recipe. Earlier this year, I had the privilege to attend the annual Prins Perinatal Society of Australia and New Zealand annual Congress in Christchurch. It was attended by over 1,000 people with presentations from all around the world. So pathology and chocolate cake, you might not think they go hand in hand, but they do, because a lot of what we do in pathology is using recipes to actually get results and answers. Anybody can make a chocolate cake, and there are key ingredients in a chocolate cake. And this study is actually about finding the key ingredients in understanding why a baby died. So a person is pregnant for 39 to 44 weeks, and we call these weeks gestations. A stillbirth is a baby who has died during pregnancy and is older than 20 weeks gestation. It is important for us to understand why a baby died, for families to grieve, to help plan for future pregnancies, and to develop prevention strategies to prevent stillbirths. But unfortunately, there are 6 stillbirth babies every day in Australia. And there are 17% of babies that we don't actually know why they have died. And this actually increases into 36% in babies that are older than 37 weeks gestation. There is currently a list of recommended investigations for us to follow as to why a baby died. There was 23 and that has been reduced now to 8. So we want to understand what were the key ingredients and help determine why a baby died. Our study went from 2013 to 2018 and involved 18 maternity services across Australia. We actually had 695 stillbirths, which is actually the second largest in the world. So we want to know which investigations or which were the key ingredients to help us determine why the baby died. In these 695 stillbirths, we found that the placenta is the most important test. It can actually help identify the cause of death in 87% of cases. The placenta is the blood and food supply that goes from mum to bub. Understanding the mother, her history, her pregnancy, and any previous pregnancies is also important in 82%. Doing a DNA test and understanding the genetics of the baby is important in 75%. And interestingly, doing some blood tests on mum looking for infections that can go from mum to bub was important in 64% of the cases. And in 47% of the cases, actually looking at the baby and doing an examination after the baby is born can help us identify the cause the baby died. So we found that the key investigations or the key ingredients are actually the placenta, maternal history, genetic testing, maternal bloods for investigation and examination of the baby. Now, as I said before, we know that 17 and 36% of babies, we don't know why they died. But now that we know what the key ingredients are, the key tests that we should be doing, can we reduce this number to as low as 5%? We currently have 6 stillbirths a day in Australia, but hopefully we can now get this number down to as low as 1 or 2. I would like to thank the wonderful people of MARTA that has actually enabled this research and will actually help us to prevent future stillbirths in the future. And to all women and families who have experienced a stillbirth, thank you.
Congratulations again to all of our winners. I have to say, as a psychologist and a mental health researcher, I was a bit nervous about some of the technical language in these bios, and I still managed to stumble on the word psychology. Anyway, we're going to break now for afternoon tea. During this break, our MARTA Foundation judging panel will be directed to room 2003 to conduct their judging process, and the rest of us will be able to enjoy some leisure time. We will be rejoining in 30 minutes. Thank you.
I would like to introduce myself again as the chair for the highlight talks. I'm Raphael Ricci, member of the neurobiology team here at MARTA Research. The highlight talk session gives higher degree research students and early career researchers a valuable opportunity to present their research work to the broader MARTA research community. AGR students, ECRs, have been nominated by their Mater Research Group leaders to present in this session, highlighting our brightest emerging research leaders of tomorrow. And I would like now to invite our first nominee, which is Dr. Sheridan Guyot. Dr. Sheridan Guyot is an experienced physiotherapist, a proud mom to 3 Gen Zs, and an enthusiastic childbirth educator, and a relatively new health services researcher. Sorry. So she leads the physio team at the Mater Mothers Hospital, where she herself was born. She's a previous Beth McGrath fellowship recipient, and her PhD research led to a plan for the Mater Mothers to support expecting and new parents as they navigate their transition through pregnancy, birth into parenthood. This real-world research is now being implemented for the next generation of Mater moms and their families. The title of her presentation is Co-Designing a Woman and Family-Centered Perinatal Educational Program. So welcome, Karen.
Thank you for having me. I just thought I would start with setting the scene with a few definitions. So perinatal, I'm using to describe the time in pregnancy, birth, and in the months following birth. And then perinatal education is therefore facilitating learning in that time, which of course is the acquiring of knowledge, the development of skills, values, beliefs, and habits related to pregnancy, birth, and parenting. How do I move the slides on? This one? For that arrow. Super, thanks. So this image here depicts a woman entering motherhood and pregnancy, and in the language of the artist who's from the Bundjalung Nation in northern New South Wales, this is a Dubai with her Jarjum. In her journey, she has a spiritual connection to her ancestors and community who will protect and guide her through the process of becoming a mother. The art depicts what Aboriginal and Torres Strait Islanders have long known, that is by supporting a mother and family through their transition to parenthood, that there's opportunity to impact the lifetime of a mother and also her infant. So if we translate that into the Australian healthcare system, maternity care recognises pregnancy and childbirth aren't illnesses, but are normal physiological life events. And perinatal healthcare for mothers brings together 3 things: health assessment to detect health challenges, treatment interventions only when they're required, and education to support the transition through pregnancy, birth, and on into the perinatal period. This women-centred perinatal education is just a core component of what is high-quality, effective maternity care that supports women and the families. So maternity health providers, the doctors, midwives, and allied health professionals aim to empower women and families through pregnancy, birth, and beyond. But at the Mater, we saw there was a gap between what education was provided and actually meeting the needs of women and their families. So my research focus has been on closing that gap at the Mater Mothers, where 1 in 5 Queenslanders are born at a Mater hospital, and with the Mater Mothers in South Brisbane being the largest maternity hospital in the Southern Hemisphere, where over 10,000 women give birth every year. So for us to effectively engage with parents through the perinatal period at the Mater Mothers, we needed a clear plan or recipe to guide that process. To achieve this, I used a relatively newly defined research methodology called implementation science. So there are 3 features of implementation science that make it a great resource to guide health service change. Firstly, it's systematic, but not prescriptive or rigid. Implementation science gives us a pragmatic or practical real-world way of embracing complexity like healthcare. Secondly, the stakeholders and their interactions within the health system are key to change rather than the processes being used. The focus is on people, on mums, families and healthcare providers, not systems and processes. And then thirdly, implementation science uses behavioural science theories to help us understand and influence human behaviour. I'm going to take you through a tour of my health service research and the local contribution it's made to delivering value-based healthcare. So the first step in developing a perinatal education program was to actually understand what was happening already on the ground and what we were missing. I used Bradshaw's model of need because it gives us a varied perspective to consider. It gave me the opportunity to identify evidence practice gaps and also identify strengths that we can build on or translate into other contexts. I studied 4 different but complementary aspects of need. The first study in my thesis investigated 2 aspects of need. Expert need, which was just comparing our current education to the evidence-based recommendations for perinatal education, statewide, nationally, and also globally. And then also express need, which was really just to observe what education we were providing and the patterns of how parents participated in that education. My second study was to look more deeply at the Martyr mothers, where we have an enthusiastic workforce. So I asked them for their suggestions and insights into how we could excel. In this study, I observed that these healthcare providers wanted to engage in the active change process. They could see some of the barriers to providing excellent education, but they weren't sure how this change could happen. I applied the implementation science framework to synthesize their responses into action points in a blueprint for success. And then finally, but most importantly, across 2 studies, I listened to parents. I asked them where they identified their learning gaps and what their expectations, experiences, and preferences for perinatal education was. I found that parents had gaps in their knowledge and skills related to important self-care topics, such as managing fatigue, important skills in relaxation and mindfulness, and also in pelvic health. And then first-time parents had unique learning gaps related to childbirth, breastfeeding, and infant care. Mothers also told me that they wanted guidance to navigate their perinatal journey. They wanted support in their transition to parenthood, but also in navigating the healthcare system. They wanted to hear from trusted voices, including their peers, and they wanted opportunity to ask questions and to learn alongside others. Ultimately, they wanted to understand themselves in the context of what is normal. Education is concentrated in the latter part of pregnancy, but women told me their learning needs were greater early in their pregnancy and after their baby was born. We're not very responsive to women's health needs, in part because we're not collecting the right data. So part of the solution is to collect a wider range of health measures that matter to women and families. So using implementation science theories, models and frameworks to synthesize the findings from these studies, I developed an outline of an evidence-based perinatal education program designed to be responsive to mothers and families. I shared my PhD findings with the Martyr Mothers team, and then we held a workshop with parent consumers, hospital leaders and maternity healthcare team members to review the findings and prioritise and fine-tune what this education program would look like. It's been a lot of work by many of the team at the Martyr Mothers, and we're now asking quality of life and other health measures that matter to women and families, and using that data to direct them to further education and follow-up care. Martyr Mothers is delivering the right education at the right time, with 12 time points to complement doctor and midwife appointments, with emails sent to parents linking them to stage-specific education, which includes infographics and short video content. Group antenatal classes are focused on skill development and questions and answers rather than on information sharing, because a lot of that information has already been shared. And then finally, the Martyr is rolling out a curated social media program where groups of mothers are cohorted depending on the month that their baby is due, and they're receiving 30 tailored posts at each stage of that perinatal journey within that group. So finally, I'd like to thank Mater Research and the Mater Foundation for their support through the Betty McGrath Fellowship that's allowed a clinician like myself to pursue a PhD and in turn contribute to the exceptional care we provide at the Mater for mothers and families who give birth there. Thank you.
Thank you very much, Sheridan, for the very interesting, important work you do. For the sake of time, we will leave the questions for the end at the networking event. And let's move on to the second talk, which is Mr. Caden Kwa. So he is a final year PhD student in Professor John Hooper's Cancer Cell Biology Laboratory. Prior to his PhD, Caden has completed his honors at the QMR and diploma in molecular biotechnology in Singapore. He has a strong interest in the development of new agents that are capable of detecting and treatment in both breast and ovarian cancers. Aside from his PhD, Caden also enjoys learning new languages and can converse in 5 different languages. That's very impressive. Also, he's audiophile, enjoys high quality playback music. Let's welcome Caden.
Thank you. Don't worry, I will deliver this presentation in English. All right, so just gonna talk about the project I'm working on for past 3 years. So advancing detection and treatment in epithelial cancers. So epithelial cancers is the most common form of cancers because epithelial cells lines all your organs, including liver, prostate, ovaries, and breast. And I'm very fortunate to be in Professor John Hooper's lab. So we works with 2 of the most lethal gynecological cancers in women. So ovarian cancer has not improved much. The 5 years overall survival has since 1975, 36%, and in 2022 is only up to 48%. And what is worse is that there is no early detection available. But whereas in breast cancer, slightly better, in 1975, the 5 years overall survival rate is 72%, and in 2022 is 92%. And this is really good because there is a worldwide effort trying to improve the patient survival. But that being said, in breast cancer, in National Breast Cancer Foundation breast cancer statistics, as you can see, in blue, the number of cases increases, but also the number of deaths also increase. Even though there is 92% survival, there are more patients dying because there is more people living now compared to back in 1975. So hence there is a need for better diagnostic or therapeutic approaches. Well, in ovarian and breast cancer, the most common way to detect is through ultrasound, CT, or MRI in the hospital, but this way you can't be sure whether you have cancer or not. This is why most doctors, they will take biopsy or do a blood test to confirm you have cancer. And from there, you either go through a cytoreductive surgery, chemotherapy, or radiation therapy. But this conventional method is not specific at all. It is very hard to differentiate between what is normal cells and what is cancer cells from us, from doctors. But even medicine as well, even chemotherapy, they can't discern well. A lot of, in conventional therapy, such as chemotherapy, when you actually treat the cells with chemotherapeutic drug, it works very well in most cancers. But what it actually does is that it targets cells dividing. Because cancer cells divide so quickly, it actually targets them. And in clinical practice, you will see tumors shrinking. But that being said, it also affects healthy cells because healthy cells also grow. And that will contribute to a lot of off-target side effects, such as nausea, vomiting, hair loss, as well as weakening of the immune system. And there is a need to actually find for more targeted therapy. So how do we actually work towards a better treatment, doing more specific? Well, in the Olympics, a lot of sports such as archery or shooting, it is that there is 3 characteristics of the athletes that they need to have in order to shoot for goal. Precision, they need to be focused, and they must have the confidence. Similarly, in cancer therapy as well, we need to have these 3 things. So how do we do it? we actually look at the difference between cancer and normal cells. But from the looks of it, there is no difference. But we scientists like to look at more molecular scale. We actually focus on distinct receptors. So these are proteins that are on the cancer cells. So when we actually know which receptors are upregulated in cancer, our lab work focuses on CDCP1. We can actually design an antibody or a protein that specifically target that to deliver your drug or agent. So what are antibodies? So antibodies are actually produced by an immune system naturally to fend off a lot of different viruses or bacterial infection, such as COVID-19. But little do you know that antibodies also attack the cancer cells, preventing cancer from proliferating. But when the immune system is actually affected, what happens is that the cancer cells just keep growing. But we scientists love to use all these natural tools to help us kill cancer cells. So by using antibodies, we know that they are specific for a specific protein. So we can actually do diagnostic and therapeutics in one package, since we know that it actually targets a certain protein that's only expressed in cancer or highly expressed in cancer. So by loading up with different molecules such as a tracer, also known as a GPS, you can actually detect with current technology that's in the clinics, such as CT, MRI. Then after which you know that the cancer do express that receptor of your interest, you can actually swap it out with a toxic drug. And this toxic drug, you have confidence that you will attack only the cancer cells and not the normal cells. So let's look at just one data that I've generated during my PhD. So on the y-axis is a cell viability. So how much cells is surviving. On the x-axis is the concentration of the drug that is treated to cells. And what is in red is an unconjugated drug, which is such as a chemotherapy drug, for example. And in blue is non-specific antibody drug conjugate. So what it means is that antibodies are specific. If I use an antibody that recognizes a different protein that doesn't express in cancer, it shouldn't attack cancer cells. And what is in black is antibody drug conjugate, which the antibody are specific to a protein that's only expressed in cancer cells. So what you see here is that in blue, it doesn't kill any cells, which is great. And what is in red, it is very toxic, and this is not what we want. But when we combine the potency of this drug to highly specific antibodies, we are confident that it actually works, and it does work. It kills cells. So here we know the antibody drug conjugate, it is safe and enriches the drug in tumors only. So with that analogy of the dude that actually won silver in Olympics, we know that this treatment is precise. We also know that it is focused. If his job is to detect, you only detect and does nothing else. If it kills, it should kill. And we have confidence that it works because it only expresses the protein that it needs. With that, I would like to acknowledge Mater Foundation and as well as all the donors that contributed to scientific research in Mater. Our group, Professor John Hooper group at Mater, MPF, over at UQ, I'm working with structural biology as well as a CMM. Thank you.
Thank you very much, Caden. Again, if you have any questions for Caden, please feel free to reach him out at drinks afterwards. So let's move on to our next speaker, which is Dr. Irina Bucko. Dr. Irina Bucko is a senior research officer in the Diabetes and Metabolism Group within the Mater Research Institute, specializing in immunology of type 1 diabetes. She completed her doctoral training in 2019 at the University of Queensland Fraser Institute, where she focused on developing immunotherapy for type 1 diabetes. This therapy is now advancing to a phase 1 clinical trial. Dr. Bucko remains dedicated to advancing treatment and prevention strategies for type 1 diabetes. Her exceptional work has earned multiple awards, and she has recently secured over $500,000 in competitive funding, including the prestigious Juvenile Diabetes Research Foundation Fellowship and Project Grant and Mater Strategic Grant for Outstanding Women. The title of her presentation is Rage Against Type 1 Diabetes. Let's welcome Dr. Irina.
Okay, thank you very much and welcome everyone. I hope the girls just had to leave and not leaving because I'm presenting. My title is Rage Against Type 1 Diabetes, and as Raphael mentioned, I work in Diabetes and Metabolism Group led by Professor Josephine Forbes, and I'm going to tell you in the next few slides how we are raging. Unfortunately, diabetes is a household name for many people, and type 1 diabetes is representing about 10% of all diabetes cases. And unfortunately, there is a few things, or actually a lot of things that we still don't know about the exact cause or trigger of this autoimmune disease, where the pancreatic cells that are producing insulin are getting progressively destroyed in these individuals. Insulin is a very important hormone that helps the sugars be transported across the body and give energy to cells. And unfortunately, when these blood sugars go high, it's not good news. Anyone can get type 1 diabetes. It does usually occur in childhood, but we need to be very aware that it's actually lots and lots of adults are living with the disease. We use a simple 4T symptom strategy, toilet, needing to frequently go to the toilet, being tired, losing rapidly weight, being thirsty, and most importantly, perhaps, is the fact that this disease cannot be cured or prevented. So once these individuals are put on insulin replacement therapy, they remain on that for the rest of their lives, whether it's daily injections or more advanced insulin pumps. And this is just a little diagram. It is a genetically, there are some genetics that are responsible for people develop type 1 diabetes, but this is diagram illustrating that actually we can no longer blame only genetics. It's a lot of environmental factors that potentially leading or contributing to the disease development, and newer and more countries sort of coming on board in the last 20 or so years. If you can imagine pancreatic health or insulin-producing health, insulin-producing cell health as a burning candle, where there needs to be a triggering event, whether it is immune-mediated or genetic or environmental trigger. And tragic thing about type 1 diabetes, that symptoms and diagnosis happen around about here, where the candle is pretty much gone. So people with established disease, their insulin-producing beta cells burnt out. So our strategies and what we are hoping to achieve in the lab is finding the way as to actually how we blow this candle out. At any point, and the earlier you try to blow it out, the harder it becomes. I did tell you that it is immunological autoimmune disorder, and there is an imbalance in type 1 diabetes between the good guys and the bad guys. I think grade 10 girls would have had these books read in the primary school, so I'm very disappointed that they don't get to see that. The good guys in the immune system are regulatory T cells. We love Tregs. We want them around, and the bad guys are effector T cells, and that looks a bit serene, but they're not good. So idea in our laboratory is to introduce the strategies to increase the good guys, make them healthy and operational. And it is actually, funnily enough, a proper immunological term, an exhausted T cell. So we do try to make these T cells exhausted. And very much like in real life, they can't perform their job appropriately, and that's our aim. The protein that we are very interested in, called Oration, it's a legitimate name, stands for Receptor for Advanced Glycation End Products. It's a very important protein in defense against bacteria and viruses. Interestingly, it's gene located in the same region that predisposes us for type 1 diabetes. It is increased in people at risk and is associated with progression to the disease. And people with type 1 diabetes have RAGE elevated in their pancreata. So we are studying a number of approaches of blocking RAGE from signaling and preventing it from damaging tissues and inducing inflammation. So we know that blocking RAGE in preclinical models improves health and function of the pancreas, and it stops type 1 diabetes development. We also look at a variety of approaches, including reduction of processed foods, and you would have heard earlier today that it's highly processed, ultra prepared foods are no good for you. And as you can imagine, it's very difficult to use that as a therapeutic, but we also know that in preclinical models, it reduces disease development and improves pancreatic health. As Raphael mentioned, I was very fortunate in obtaining Mater Foundation funded strategic grant, and we are looking at very novel approaches to blocking rage in a very novel systems. Another way we are exploring at the moment is using a tablet, which is very easy and friendly way of delivering a therapeutic to individuals with type 1 diabetes. And again, we see that this particular therapeutic reduces T1D development, improves pancreatic health, and increases the good guys whilst making the bad guys exhausted. And most recent and the most exciting venture of ours is a collaboration between Mater Young Adults Health Centre and Queensland Children's, where we are embarking on a huge journey of search for a needle in a haystack, where we are hoping to investigate rage, behaviour and signature in blood of young children at risk of developing type 1 diabetes or newly diagnosed, and that is within 100 days of diagnosing type 1 diabetes. So we are pretty confident the rage is going to be, the rage's behavior and signature will take us further down the track as to how we can prevent and stop disease from developing further. And with that, I'd like to thank my team and led by Professor Josephine Forbes, who is our main rager, and of course, Mater Foundation for allowing us to do the work that we do, and without the support we won't be able to. And I thank you all, and I hope I didn't make you exhausted.
Thank you very much, Irina. If you have any questions for Irina, just come find her afterwards at the networking event. So let's go now to our final talk for the session. That is Dr. Jasmine Antoine, is that correct? So Dr. Jasmine is a neonatologist at the Mater Mothers and Neonatal Clinic Critical Care Unit. She also works as a retrieval coordinator for the newborn retrieval services in Southeast Queensland, which is Neo Rescue. Jasmine's interest in education, simulation, and improving clinician and patient's experience led to her PhD. Her research is aimed at improving the understanding and training of complex tasks, in particular newborn intubation. Jasmine was awarded a Betty McGrath Education Seating Grant in 2023. So the title of her talk is Mastering Newborn Incubation. So let's welcome Jasmine.
Good afternoon, everybody. A Formula One race car takes less than 3 seconds to complete their pit stop. And in that time, a team of very highly skilled crew pit members complete multiple tasks. They work in unison, changing the tyres, refueling the car, and much more. The processes of a pit stop are quite similar to newborn intubation. Takes a little longer than 3 seconds, somewhere between 30 and 90 seconds, but we have a team of highly skilled clinicians, so both doctors and nurses, working together in unison to complete the task of intubation. And it's complex, it's time pressured, it's high stakes and difficult. Newborn intubation is complex because it involves multiple different steps, and these steps need to be performed in a particular order. It also involves the potential of multiple different outcomes. So the intubation may be successful, it may be unsuccessful, the baby may become unstable during the process. Newborn intubation is time pressured. We know that when babies need to be intubated, it needs to occur quickly and it needs to occur on the first attempt. And this is in order to keep them as stable as possible. We know that newborn intubation is high stakes. Babies are very fragile, and the rates of complication associated with intubation occur in about 1 in 2 babies. So that might be that the breathing tube is placed in the wrong spot, that it causes damaging to the surrounding tissue, or that the baby becomes unstable during the process. And newborn intubation is difficult. So we intubate babies as small as under 400 grams. And as Brona told us earlier, babies are not just small adults, so they're incredibly small and they have small airways. The problem is that we know that if you're a senior doctor, you're far more likely to have success at intubation, probably around 90% on your first attempt, than if you're a junior doctor. So junior doctors' success rate is about 50%. But the problem is that senior doctors are not always available. Babies need to be intubated at all different types of day, all days of the week. So how do we train junior doctors to become as successful as senior doctors? Because it's hard to learn to intubate. Across Australia, 4,500 babies need to be intubated every year, and the Mater is the largest maternity hospital in Australia, and we intubate about 350 babies. So this is an important problem for us to find a solution to. It's hard for junior doctors to learn to intubate because not only do they need to learn the skills to be able to intubate, they need to be able to retain those skills over time, even if they're not performing it very frequently, even if it's only once a year, they need to still be successful. And they also need to have flexible skills. And by that, I mean that they need to be able to intubate any size baby in any location. So that might be the birth suite, the operating theatre, or the neonatal critical care unit, and with any different team members. We know that junior doctors becoming more successful at newborn intubation is not just achieved by training them more. Just like in Formula One, the success of the pit stop is not just reliant on the highly skilled team of crew members. It's also reliant on having the right processes. So you can't change the tyres on a car unless you first jack it up off the ground. And it's reliant on having the right equipment for that environment. So having a car jack that takes 30 seconds, say, to get it to the maximum height to change the tyres is not helpful in Formula One when you need to be in and out of that pit stop in 3 seconds. So too in newborn intubation, it's really important that clinicians gain skills and their team members gain skills, and that we have the right processes with the right equipment for the environments that we work in. My research is focused on a 4-part solution. And firstly, we've reviewed the current training literature to see, what's the most effective forms of training and efficient ways to train people. We're also identifying the important parts of intubation so we can change those processes and improve them. And we're testing new types of training. So we're able to develop clinician skills that they're not only able to retain over longer periods of time, but also that are flexible. So they're as successful in those different environments. You're as successful when you have a whole new team working together. We're also testing new equipment to find what works best in our environments. Our aim is to improve the skills of clinicians, to improve the processes of newborn intubation, and also to improve the equipment that we use. And we believe that with these changes, we'll be able to help clinicians to master newborn intubation so that we have higher rates of success on the first attempt at intubation and we have fewer complications. Thus, we'll have safer intubations for babies, no matter the availability of senior doctors. I'd like to take this opportunity to thank the team that I work with, my ETT notes, and also my primary supervisor, Professor Helen Lively, and also thank the donors and supporters of Mater Foundation and Mater Research. Thank you.
Thank you very much, Jasmine. If you have any questions for Jasmine or any other of the speakers for this section, at this last section, please feel free to ask them questions at the drinks. And I really encourage you to do to ask them a lot of questions. And with that said, I'll hand over to Professor Allison to announce the ECR seating grant winners.
Thanks, Raphael. We are really excited to be able to offer a seeding grant to our early career researchers to really help accelerate, thank you, to really help accelerate their career path and allow some really innovative research to be done. And of course, we wouldn't be able to offer these this opportunity to them without the support of the Mater Foundation, but also, just as importantly, some support of consumers who help to inform our research, and even more so now are actually involved in helping to develop the research questions and the approaches to our research, and that's really important. And hence why, as part of this process, we now engage a donor and consumer panel to help us make our final decision with respect to the ECR seeding grants that are awarded. And as per every other time I've been involved in this, I was amazed at the engagement of our donors and consumers, how well informed they were about the research due to the really excellent presentations that were given by our finalists and all the information that we provided to them. And so it was a really robust discussion. There were different viewpoints, and it was fantastic to be involved in. And as always, it's an incredibly difficult decision in the end, because each one of these projects really deserves to be funded. So I'd really like to thank our donor and consumer panel, that is Miss Liz Coughlin, our new member of our Health Consumer Network, David Young, and also a student from the All Hallows School, Miss Aislinn Wood, and also another supporter of Mater Foundation, Nita Buckingham. So I really appreciate your time and contributions to making this decision. And if I can ask both Liz and David actually to come down to the stage here to help present the finalist certificates as well as the winner certificate, that would be fantastic. And so we have, because of the scope of research we have at Mater Research, we have grants for both biomedical and clinical research. And in the clinical research category, I'm very pleased to announce that the winner of this year's ECR seeding grant is Brona McAllenden. You can please come up and get your certificate. And also, if I can ask Hamid to come down and also collect the certificate as well. This, again, like I said, was a really hard decision. And, you know, I think it's great that we have such high quality research being proposed by our early career researchers. And just want to continue to encourage the unsuccessful applicants, that research resilience and persistence is absolutely key. So keep trying, because we're confident that your research will get funded in some way or another if you keep persisting with it. So now if I could invite Nita, and it is going to just be Nita because Aislinn's had to leave, to come down. So thank you, Liz and David. You can go back to your seats. Much appreciated. So to announce the winner of the biomedical ECR seeding grant, I'd like to congratulate Dr. Charles Bell. So again, it was an incredibly tough decision by the panel, especially given the diversity of research that we have to judge. And again, to the other finalists, who, if you can also come down, Stephanie and Raphael, to get a certificate. We encourage you to continue to persist to get your work supported, because it's very deserving. Now I have the opportunity to hand over to Professor Brian Gabrielli. And he's going to present, I think, the David Ceruzia. We've already done that one, haven't we? Yes. But Brian, I think you get to do the closing speech, right? No? See, I cannot follow instructions. Never give me instructions. We're handing over to Paul Dawson, who is our head of education and so has a lot of responsibility for the carriage of our student MARTA research and does an absolutely fabulous job of it. Thanks.
Great, thanks, Ali, and good afternoon, everyone. Before we move out to the atrium for the opportunity to network over food and drinks, it really is my privilege to close this event by thanking all of our guests, our staff, our students, presenters, session chairs, and all of those people that have worked really hard behind the scenes for today's event. I'd also like to say a special thank you to Raya and Dr. Jacob Gunn for their patient-clinician duet. And so the Professor John Prins Oration is really an important part of this event. It reminds us all of the reason why we do research, and that is to help patients. And so thanks very much to Raya and to Jake. And on behalf of all of the MARTA research team, I'd like to thank all of the philanthropic supporters that are here with us, and also for those that have had to leave early for their help, who help further and accelerate life-changing research. A few who are joining us today include Liz Coughlin, Nita Buckingham with the MARTA Hospital Brisbane Auxiliary, our consumer advocates, David Young and Kathy Shoemaker, and All Hallows Schools, who attended with their Dean of Teaching and Learning, Mr. Scott Adamson. And so now I'd like to invite everyone to join us for drinks and networking outside. But I'd just like to remind all of the judges, the MARTA Research Executive, our All Hallows School team have left and already had their photos taken, so that's okay. Members of the advisory board that are still with us, Professor Denise Doolan, and I'm not sure if Dr. Kari Hilliard is still here, thank you, if you can remain here. The John Prins Oration speakers, I know Jake is still with us, I think Ray has had to leave. UQ executive, our winners, finalists, session chairs, if you can please remain here at the front of the auditorium for some special photos. And if anyone's got any questions for our presenters, please approach them outside over drinks. Thank you very much. I look forward to another successful Future Leaders Symposium next year in 2025. Thank you.